As technology advances, it revolutionizes the world with new and exciting applications, but ethical issues arise with each advancement. A great example of this is the rising field of genome editing. A genome is the sum of genetic material present in a cell or an organism. Genome editing, then, is the process in which DNA is inserted, deleted, modified, or replaced in the genome. Genome editing is a field that is relatively new and rapidly advancing. Targeted genome editing was preceded by the understanding of genetic mutations. In the early 1900s, it was discovered that mutations occur randomly and that mutations could be induced by radiation and chemical treatment. Later, in the 1940s, it was discovered that modifications can be made by transposable elements or segments of DNA that jump around in the genome. The problem with these methods is that mutations are all random. It wasn't until the late 1900s and early 2000s that targeted genomic changes became possible. Instead of random mutations, modern technologies allow directed mutations in most cell types and organisms. The newest and most utilized technology is CRISPR. CRISPR stands for Clustered Regularly Interspaced Palindromic Repeats. The technology is based on a natural viral defense system found in bacteria. There are two main steps in which CRISPR works. First, a guide RNA molecule directs a Cas9 protein through the genome to a location that matches the RNA. The Cas9 protein binds to the DNA at this spot and acts as a pair of molecular scissors that cuts this portion of DNA out of the genome. Naturally, the cell will try to repair the broken DNA, but this is where genome editing takes place. Second, scientists can insert a strand of DNA that they previously designed with any mutations they desire, and it will incorporate itself into the genome. With control of the guide RNA and the new strand of DNA, scientists can modify essentially any part of the genome any way they desire. Editing the genome allows for changes in an organism's phenotype, including changes in appearance and internal functions of the organism. This technology is revolutionary because it allows for very precise editing of the genome, opening up the door for countless applications. One of the biggest applications is the prevention and treatment of disease. Many genetic diseases are caused by the mutation of a single nucleotide base. CRISPR has the capability of editing diseased cells by fixing the mutation and restoring normal function. Examples of diseases that could be corrected in this way include cystic fibrosis and beta thalassemia. Correcting these diseases in the germline, or sex cells, allows for the prevention of disease, not only in the edited generation, but also in future generations. This process has been done successfully, but most research is still being conducted in mice. However, there is huge potential for simple correction and treatment of genetic diseases in humans. Another application is the study of gene function. One of the best ways that researchers can study gene function is by looking at the effects of mutations on genes. CRISPR allows researchers to make any mutations they desire, making the study of gene function a lot easier. This can lead to better understanding of diseases, providing even more potential for disease treatment. Additionally, CRISPR technology has potential for agricultural innovation as well as many other non-human applications, such as editing mosquitoes so they can't spread malaria or the Zika virus. The CRISPR technology is very new and exciting and is only going to develop more quickly, introducing more and more applications that could revolutionize the world. It is very important to be aware of this technology because it has great potential to change the lives of everyone in one way or another. The downside is that all sorts of ethical issues come.
Let's discuss some of the ethical concerns related to genome editing. Most of the ethical issues with CRISPR are not about what it has done, but rather what it has the potential of doing. According to Rodriguez E., there is great concern regarding the effects of genome editing in the human germline, since the edited genomes in the germline can possibly be carried down to future generations. Since genome editing via CRISPR has shown to produce mutations and side effects in human cells, it's possible for unpredictable changes to be transmitted to these future generations. Those against genome editing argue that this is a violation of informed consent. Future generations have no way of giving their consent to possibly inheriting these unpredictable effects, simply because they do not exist yet. Even if they could, there would still be no way for them to know what they would be consenting to, since the effects would be unpredictable. The counter-argument to this is that since genome editing in the germline is currently unsafe, genome editing technology needs to be further researched and developed to be made safe. Another ethical dilemma associated with CRISPR is the use of genome editing for non-therapeutic intentions. This includes what are called designer babies. Sarah Lai defines a designer baby as a baby genetically engineered for specially selected traits, which can vary from lowered disease risk to gender selection. Ethically, there is a concern regarding economic and social standing. If technology like CRISPR becomes accessible, then only the wealthy will be able to financially afford editing the genomes of their children. This means that money will guide who has and who doesn't have favorable genes. Furthermore, there will be social distinctions separating those who have been genetically modified from those who haven't. Those in favor of the idea of designer babies argue that parents already heavily influence the outcome of their children's lives through environmental choices. For example, parents who want their child to be an athlete will sign them up for sports classes or encourage them to join a sports team from their school. And parents who want their child to be a musician will sign them up for music classes or take them to concerts. Additionally, genome editing technology, such as CRISPR, could be used to control the actions and behavior of certain people. To be more precise, it may be possible for the criminal justice system to force perpetrators of violent crimes to have their genomes edited to decrease violent behavior. This begs the question of how this type of genome editing can affect these criminals for the rest of their lives. While they may no longer be regarded as criminals, the degree of how much their violent behavior is reduced is concerning. Depending on how heavily the genomes are edited, the criminals may completely lose their ability to express anger or feel anger. Some see this as forcibly taking away a person's ability to feel and act a certain way that they should naturally be able to express. By taking away or reducing the criminal's ability to feel anger, they have basically had something that made them normal human beings taken away from them. Those in favor of genome editing argue that this type of behavior modifying can be used to help people overcome their addictions, and they also argue that this will prevent criminals from committing violent acts repeatedly.
The potential scientific application surrounding the CRISPR Cas9 genome editing system has driven countries into another scientific Cold War, with each country aiming to be the first in mastering genome editing in human systems. China currently leads the race, being the first country to successfully use CRISPR on human embryos in 2015. However, human embryos were not the first step for China, as they had already accomplished various trials with CRISPR Cas9 on animals of all variety, including beagles who were targeted to overexpress muscle production and pigs who develop organs which could potentially be used for human transplants. The United States, not to be outdone, was cited in a recent National Institute of Health publication promising $190 million in research grants for somatic cell gene editing treatments over the next 6 years. This aid comes with the hope of fast-tracking CRISPR-Cas9 editing systems, making them easily accessible to operate in human clinical trials against disease. Big spending on genomic research is nothing new to developed countries across the globe. In fact, the U.S. spent $4 billion over the course of 5 years to sequence a single genome from 1988 to 2003, while the British government embarked on a $0.4 billion 3-year project to sequence 100,000 whole genomes. However, while these developed nations are making scientific strides with significant price tags, the gap between medical prosperity in developed and underdeveloped countries is only widening. To low and middle income countries across the globe, the promises of the CRISPR-Cas9 system seem elusive, as they currently struggle to receive the most basic form of medical care. Ironically, these countries that are considered low-middle income account for 90% of the global burden of disease, yet only attribute to 12% of global public health spending. A recent study by the Johns Hopkins Bloomberg School of Public Health discussed the inequality and inefficiency of healthcare services in low-middle income countries. These communities have lower numbers of health workers, drained medical stocks, and higher costs. In fact, 60% of individuals pay out of pocket for medical services, making a routine checkup a formidable and costly task. Cost isn't the only deterrent, however, as most public clinics are miles away, understaffed, and have inconvenient hours of operations. Thus, individuals in poverty fail to receive proper care, either by their inability to afford it or the clinic's inability to provide proper aid. This only builds into a cyclic pattern of public health, as those in poverty fail to receive care and thus are more likely to die from illness, which in turn takes away from the gross income of a country and results in increased healthcare costs per person. With this in mind, the current craze of curing illness through CRISPR Cas9 in these countries is nothing short of a myth, as even if clinical research was allowed to occur within the communities, it is unlikely that patients will ever be able to afford these genetic treatments with costs in the thousands. This expands the moral arguments over CRISPR Cas9 far beyond the terms of scientific justification, but also into the further minimization of impoverished communities, as those who would benefit most from genetic therapy and account for a majority of the world's disease will not be on the receiving end, but rather watching from the sidelines as another life-saving medical advancement falls out of reach. While medical communities of developed nations are waging arguments to mainstream CRISPR Cas9, impoverished communities only continue their fight to afford basic health care. If the new age of human medicine is so rapidly approaching, will only the wealthy be included in the new genetic elite, while those who can't afford treatment remain in the past? With science on the brink of mastering CRISPR Cas9 in human patients, these questions will need to be answered before humankind becomes built by design.